Hey guys, hope you guys are doing well. So for today's session, I'm gonna start off with two questions. If this lets me, okay. So the question is asking this. Hold on, let me just move this here. In the following coding strand of DNA, exons are colored in black and introns are colored in red. And so this is our sequence in the coding strand. What is the sequence of the complementary pre-mRNA? So what do we know in this question? We know that it is the coding strand. So I forgot to mention this in the last session that we did. So we have a coding strand and a template strand. I'm going to show you two different questions of this. So don't worry. So when it's the coding strand, we should know that to get the pre-mRNA of the coding strand, you just need to change the T's to the U's, right? With RNA having uracil instead of thymine, right? So when you do that, so you have this sequence, right? We have these guys here that are introns, right? The red introns. So when it's pre-mRNA, you keep it at, when you transcribe it, it comes down to this as the pre-mRNA sequence. So you're just changing the T's here to a U. This is before anything splicing occurs. This is the pre-mRNA sequence. Okay. Now this is another type of question where you would use the template strand. So consider the following segment of DNA and assume that the top strand is the template strand used by RNA polymerase. Draw the RNA transcribed, label its 5' end and 3' ends. Okay, so what do we know? We know that the top strand here, this guy over here, the one that says 5' here, is the template strand. When you hear template strand, you always end up using the complementary strand. So if this strand wasn't given already, you would take the complementary of that template strand and then you would transcribe it by a, so you would take that complementary strand and then change the T's to the U on the complementary strand or the coding strand. So this is now the coding strand, right? Template strand, coding strand. You transcribe the coding strand. That's how you get this sequence right there. Does that make sense? I hope it does, because this kind of leads into what we need to talk about today. So just to recap, the central dogma of biology, DNA to RNA to protein. Always remember that. Don't ever forget it. It'll be your best friend forever. So hopefully you guys have ingrained it in your brains as of now. So today's focus is more on protein and how protein is made, right? So we're going to focus on the latter half of the central dogma. So before we talk about translation and turning it into protein, let's actually talk about protein. So protein structure, what is it made of? It has amino acids being the building blocks, where you have an alpha carbon, the central alpha carbon, an amino group, a carboxyl group, a hydrogen, and the variant R group. The R group determines the type of amino acid that it is. So when you look at it, it can either be nonpolar or polar. If it's nonpolar, it's typically hydrophobic. If it's polar, it's hydrophilic. Depending on the case, it can be further divided into non-charged, positively charged, and negatively charged, okay? So these are the different amino acids. I don't think Dr. Bruce said he wanted it. I don't know how in depth he wanted you guys to go with the amino acid structures, but just know that these are the different classifications: nonpolar, positively charged, negatively charged, polar. So these are the main groups in there. So when protein forms, it's by joining the carboxyl group of one amino acid, and you take the amino group from the second amino acid, and you kind of take that together, you dehydrate it, so you take out any water, so you take out an oxygen and two hydrogens, you bring them together and be like, ooh, yay, we're now together, we're building things together. And that bond that forms between the carboxyl group and the amino group here is called a peptide bond. So that is the basic of how a peptide bond is formed right there. This is one of the basic structures of a protein. So in the primary structure itself, you see it as a sequence, right? So it's a sequence of amino acids in a polypeptide chain. So remember how we had one of our, from our previous example, we saw that we had one forming a peptide bond with each other, right? So poly just means many, right? So many peptides. So you have all of those joining together, all of these amino acids coming together and joining by peptide bonds. So when this is just a fact, how you would name or number the system itself, it always starts from the N terminus, and you would name each of the amino acids starting from the N terminus towards the C terminus. The C terminus is the end of it. N terminus is typically marks the beginning. Okay, so the side chains are not part of the backbone. And so the backbone of a polypeptide chain is the nitrogen, the amino term, the N terminus, alpha carbon, and the carboxyl carbon, okay? So the nitrogen and amino group, alpha carbon, and the carboxyl carbon. That's part of the backbone. So you kind of see it in the middle here, right? You see it there. You kind of go on between each of these guys, ooh, my bad. So you see that it's nitrogen, carbon alpha, carboxyl carbon, amino nitrogen, carbon alpha, carboxyl group. So that's the backbone. These side chains are always going to be more out towards, it's not part of the backbone, it's facing out of it, right? Okay. So now the secondary structure. This is where the different side chains kind of come into play. Wait, no, my apologies. Not the different side chains. The carbonyl, so that carbonyl oxygens, the C double bond O's, and the hydrogens in the amino groups actually form hydrogen bonds between each other. That's how the secondary structure ends up forming. And it typically has 3.6 residues per turn, give or take. So that's just kind of this is kind of an alpha helix. So typically a secondary structure, you have it as either an alpha helix or a beta pleated sheet. It's the same thing, it's just one thing, just hydrogen bonding with each other, right? So in this you see the hydrogen bonds going between the oxygen and the hydrogen here. Same thing over here, just in a different shape, okay? So in this case, yep, same thing happening. So hydrogen bonds formed between the carbonyl oxygens and amino hydrogens of the backbone. And it involves 2 or more beta strands to become to form pleated sheets. So these are one alone. If you have multiple of those guys, then you end up with a sheet, okay? So tertiary structures, they involve nonpolar residues towards the interior of the protein. And the interactions become many among the side chains. So the side chain, it plays an essential role in forming that tertiary structure. Now we have the quaternary structure, which is pretty much with nonpolar residues going in the interface between polypeptides. So essentially you have multiple polypeptides, they come together, they kind of keep the nonpolar. So remember what we said about nonpolar amino acids. They are hydrophobic, right? So when you have a hydrophobic molecule, it tends to go inside, right? So we have our nonpolar ones in the inside. This is another one. It comes in. We kind of keep the nonpolar residues inside with the polar ones that can interact with the environment outside, okay? So again, the side chains play an important role in this matter because depending on whether the polarity is, how the polarity is, that kind of changes where the position of that polypeptide can end up in structure wise, okay? So the next thing would be, what is a functional domain? So a functional domain is essentially a sequence with 50 to 300 amino acids, okay? So those are like, so these guys here, it's around 50 to 300 amino acids. Those are called protein domains or functional domains. In this case, sequences that constitute protein domains, they fold into stable, unique conformations, and these different domains end up giving different functional capabilities. So with PAS, this guy here, it binds siRNA and microRNA. Here we see hold on. We see this guy here where it's binding small non-coding RNA, and then this domain here ends up doing RNA hydrolysis. So it depends on what the domain needs, what is going on, and we kind of just go from there, okay? So that is a functional domain. So this is a video. I'm not going to show it here just because the audio itself does not like to work in this thing for me. So I will put the link to this. Make sure you guys watch it. It will help you a lot. So now the next section itself is going to be more like a question kind of thing. So when you guys go through this next section, try to answer the questions before you kind of go through it, okay? So what is translation? And what does it require? The process, so translation is the process of converting information on an mRNA molecule into a sequence of amino acids that make up proteins. It requires ribosomes, tRNAs, amino acids, and energy. So in this case, we have DNA going to mRNA. That was our transcription part, right? Then we convert that into mature RNA, which is our RNA processing step. Now we're going to talk more about how this actually occurs with transporting it in. And so the mature RNA ends up getting transported into the cytoplasm, and it eventually forms that it forms the polypeptide chains or the proteins themselves, right? So what does the ribosome do? It provides the scaffolding on which the tRNA or mRNA are brought together, and it allows the formation of peptide bonds. So it holds the tRNA and it holds the mRNA and it brings in the tRNA, so it has a place to stay while this peptide bond formation occurs. So here in prokaryotes, you have a 70S chromosome with different units here, the 50S subunit, 30S subunit, these guys here, the different RNA, these are ribosomal RNAs. So 5S rRNA, okay? And then we have the 30S subunit, which is the smaller unit on this guy, and it has this guy. So so these are different rRNAs on the group. These are the two components. And then with eukaryotes, you see the differences with how the subunits themselves are. So it kind of brings them all together. So different structures means it's to accommodate the different needs for a prokaryote versus a eukaryote, okay? So how do tRNAs decode the mRNA template? So the tRNA itself has a specific amino acid that attaches it to its 3' end. So here we have so in mRNA, when you have the different nucleotides come in, these guys come in, the tRNAs come in, this guy, and so we have something called codons. It is 3 nucleotides each for RNA. So each codon is 3, it's a triplet codon. So in this case, you see that GCA will bind to CGU. This guy comes, so this part here on the tRNA is called the anticodon. And that anticodon comes to bind to the codon because it's bringing the amino acid that is bound to the 3' end up here, okay? So this 3' end of this RNA is where the amino acid is actually attached. And so just general understanding of tRNAs, they fold into a clover leaf shape. It is the secondary structure of the RNA itself. And the anticodon loop is what defines what codon it decodes. So this anticodon here, this loop here, determines which guy comes in here. We'll talk more about this when we actually get to the genetic code part of this session. So essentially with this guy, it kind of just matches what is happening here, okay? So in this case, you have GCA matching with CGU. CGU is for the amino acid alanine, right? So this is a question. You guys should try this on your own first. Pause it here and then we'll go from there. So label the diagram with the following translational components. Ribosome, large and small subunit, the growing peptide, polypeptide chain, amino acid attachment to tRNA, mRNA, codon, anticodon, okay? So that is our question. So where is the ribosome? So we have the small ribosome here, right there. We have the large one there. We have the polypeptide chain up there with the little balls, little round balls going on, circles. Then this line here, you can see it's the mRNA trying to pass through. It's typically through the small ribosomal subunit that the mRNA goes through. The tRNA is coming on from here, this clover leaf shape. It comes in, it's there. you have an amino acid attached to it. You have the codon down there. So codon, remember, it's always in the mRNA. Anticodon is in the tRNA. Ooh, sorry. I hope that makes sense, just understanding what is where, how that kind of goes. Now so we have 3 stages in translation: initiation, elongation, and termination. So so what actually occurs in initiation? So in initiation, this is where the main difference between prokaryotes and eukaryotes come in. And so with prokaryotes, the initiation complex comes in. It has the small ribosomal subunit, the initiation factors, and the mRNA ends up binding near this start codon area, right? So before the start codon comes in, there is a sequence called the Shine-Dalgarno sequence. This is where the mRNA comes and sits in the ribosome, okay? So that sequence pairs with the 16S ribosomal RNA, and it allows initiation to begin. In eukaryotes, there is the difference where you see that it recognizes this 5' cap or 7-methylguanosine, this guy here. So when you see, so the small ribosomal unit comes in, binds to that side, it comes in, it's like, ooh, scanning, scanning, scanning. Yay! We see the start codon. Woo! And it's like the start codon, recognizing that, it kind of brings in what is called methionine, or the start codon, right? AUG is our codon for starting, and then everything kind of initiates that. There are a couple of other factors that are involved, but this is the main thing that comes into play to get everything started off. So what happens in elongation? So elongation, as it says, it's kind of just bringing all the amino acids in, right? So it's adding them one by one to that polypeptide chain. So it starts in the amino acyl state. It binds, so it brings in the amino acyl tRNAs, so the ones that have the different polypeptides here, right? So it brings it in, it shifts over a bit, and then it's like, ooh, next. It's like goes in, so it comes in, and then it goes into the P site, which is the peptidyl site. This is essentially where the peptide bond forms. So that kind of goes in, it goes in, it adds the next one on there, shifts it, adds it. This guy gets shifted, this shifts over and exits out. So the E site is just exit. So So that kind of continues until they reach a stop codon or a release factor, depending on the situation or the circumstance. So what occurs in termination? As the name says, it stops. So elongation is just adding it on. The moment it finally finishes is when that RNA reaches a stop codon. So when you have a stop codon, you just have a release factor that binds to it, and it's like, oh, yay, free RNA going on here. And then you have the free polypeptide, and then everything just disassembles because the release factor is in there. So everything is free, we are all good, and it's happy. So stop codons are also called nonsense codons, UAA, UAG, and UGA. So whenever those are encountered, you end up with a stop. You terminate everything. You're like, the ribosome's like, okay, we're done. We're good. Rip. Unless there's more signals to keep the process going. Let me clarify that actually. So this will, so for this specific protein, it's done. It will start the process again through initiation if there is more required, but that is a story for another day when we talk about gene regulation and all that fun stuff. Okay, so the release factor just binds to the stop codon and we're all happy, right? It's everything's free, we're all good. So this is what I was talking about with the genetic code. So the genetic DNA codes for protein in a triplet code. It is always read through 5' to 3'. It's non-overlapping and contiguous. Universal. It's universal with the exceptions of organelles, mostly mitochondria and certain protozoans. This genetic code itself is degenerate, which means that several codons call for the same amino acid, and 61 codons actually code for 20 of these guys. Mutations, depending on the case, there are point mutations. There are silent ones where no substitution occurs. Missense ones, amino acid substitution actually occurs. Nonsense ones where it ends up in a stop codon. Then there are frameships where a certain inserting or deleting a certain number of nucleotides end up with a complete change in the amino acid sequence itself. So the number of nucleotides is typically not divisible by 3 in that case. So it kind of just shifts everything, right? It either shifts it down or shifts it. It kind of just changes depending on the case. So in this question, a glycine residue is in position 210 of the tryptophan synthetase enzyme of wild type E. coli. Use the codon chart to answer the questions below. If the codon specifying glycine is GGA, how many single base substitutions will result in an amino acid change at position 210. So to answer this question, you would end up listing out how many different combinations you can have. So each spot has 3 other choices that can happen, right? So in this case, you end up with 6 different changes that you can make, and each of them gets 3 each with that. That gives you an amino acid change. There are a couple of other options where it changes the A. So you have GGA, right? Any change in that last position only does not change the amino acid. It is only glycine, right? Because any other change ends up changing it to a complete different amino acid. Like so we have GGA. If you have UGA, so the first position you have U, second position G, and then you look for A. Oh, wait, sorry. So U, we go here to G, and then you see UGA, which is a stop codon, right? So that's how you would look at the genetic code table. I should have went over this earlier. So the first position, second position, third position. That is how you would go about it. So one position here, first position, so U, G, second position, G, and then you would look at that section here where that A is, that's where you get UGA. So this guy, we know that it's a stop codon, right? So that's how you kind of continue into asking this question, which amino acids are actually getting involved if a single base substitution occurs. You have this guy in there and be like, oh, so UGA is this guy, these guys are arginine, this guy is alanine, these two are glutamate and valine. So that is kind of how you would understand how that would go, right? So consider the following segment of DNA and assume the top strand is the template strand and used by the RNA polymerase. Draw the RNA transcribed, draw the corresponding amino acid chain, and label its amino and carboxyl ends. So in this case, you would start off. So remember how I said with template strands, right? I took this question from earlier and we transcribed it to this guy, right? So now what we need to do is use our genetic code table and figure out which one is what. So we have UG. That gives us leucine, right? We have leucine. GGA, which is this guy here, that's glycine. AGC, so A, G, C, which is serine. And then remember when we always say the 5' end is our N terminus, the 3' end is our carboxyl end. So that's where it gets added on, okay? Let's do another question. So we're going to do this question where given the following DNA coding strand sequence, what is the polypeptide chain produced? So in this case, we transcribe it. We have a coding strand, right? So since we have this coding strand, we only have to change the T's to a U, and we end up with our our mRNA strand for this guy. And then you want to translate it. When you translate it, you see AUG is methionine, right? AUG, our wonderful start codon that we love. And then we have UUC, that is phenylalanine. Then we have proline, which is CCC, which is proline. Then we have AAA, which is right here, which is lysine. And then that's where that ends with that case, because UGA here is a stop codon, right? I hope that makes sense so far. So let's do another different question. So a protein is 300 amino acids long. Which of the following could be the number of nucleotides in the section of a DNA that codes for this protein? So we know that 3 nucleotides equals 1 amino acid. 300 amino acids equals 3 nucleotides times 300 amino acids equals 900 nucleotides in a single strand. But in this case, we have to remember that DNA is double-stranded. So the number of nucleotides is twice the number, right? So 900 times 2, that is how we end up with 1800 nucleotides, right? So the primary structure of a protein is determined by what? The sequence of amino acids, right? Yeah, I know you guys are awesome. You guys better be looking through this and be like, yeah, I know this. So what's this guy? The secondary structure of the protein includes what? No, you guys know this. Alpha helix and beta pleated sheets, right? Good. How are protein structure and function determined? So protein structure is determined by the sequence. Protein function is determined by the structure itself. So the sequence determines the structure determines the function. Okay? So, okay. A tRNA in E. coli charged with aspartate would have which anticodon? So I know with Dr. Bruce, he loves questions on exams where he would go about trying to figure out which one, what is the sequence for, or a specific set of amino acids bond together. So like if you have a protein, he'll give you the sequence of the amino acids and then give you this guy, right? Like be like, which one in here is what, which is the sequence for that specific sequence, right? So in this case, we're that's a similar case, except this is just showing one amino acid, and it's asking which one is the anticodon. So the anticodon is binding to whatever is in this guy, right? So we have aspartate. Where are you? Oh, there you are. Okay. So now we need to see which one binds to this guy. It's either C CUA, or we have, oh, wait, where did the mouse go? Oh, there you are. CUG, okay? So remember, we always read from 5' to 3'. So the answer here is this guy, right? See how 4 is not the answer because the C would have been the 3' end, right? Okay. So CUG, when I said it, that was from 3' to 5', but make sure you guys always switch it to 5' to 3', okay? How was it demonstrated that the genetic code consists of triplet nucleotides? So so this was conducted by an experiment where they inserted and deleted 3 nucleotides. When they removed 1 or 2, they actually saw that it kind of shifted everything, so it didn't preserve the function of that resulting protein itself. So that is why the answer is C. I don't think you need to know how they got to it, but if anyone asks, that's how it happened. An insertion or deletion of 3 nucleotides within the coding region actually preserved the function of the resulting protein. Okay. Suppose you discover a new bacterial species which utilizes codons consisting of 2 nucleotides each, and that only one of its codons function as a termination codon. How many different amino acids can be encoded by this genetic code? The answer in this case is 15. Because it's like the 2 to the n case. Oh no, wait. 4 to the n, sorry. I will write this down for you guys in the PowerPoint itself, but it's 4 to the n, where n is the number of nucleotides. So in this case, 4 to the n, 4 to the second power, which is 16. But you have to remember that here we have a codon that functions as a termination codon. So that doesn't, remember with termination codons, you do not have an amino acid coded for it. So in this case, you end up with 15 because you have 16 codons in total, and you end up with it, the codon itself, 15 codons. So 15 codons and a stop codon. And so let me just write that out for you guys so you guys can see this much better. Okay. I will write that down later so you guys have it on record just in case if a question comes up. Let me know if you guys have any questions. This is pretty much what I have for today. Okay, if you guys have any questions or concerns, let me know. I know this is a little bit last minute that I'm uploading this guy. So hopefully you guys get a chance to look through it, work through the problems, and yeah. All of you guys stay safe, okay? Miss being in class with you guys, but I hope this is helping you guys. Let me know if anything isn't helping because this is all new to me too. So let me know if these videos are helping or not, or any concerns or suggestions. I'm always here trying to take your guys' opinions to better improve, helping you guys get through this. So I will see you guys next time if you guys don't have any questions. If you have any questions, email me, whatever you guys want. Sayonara. Bye bye.